So let's talk about chapter 2, the chemical level of organization. Um, a lot of students, there's a little bit of fear with chemistry. This is not a chemistry class. I'm not trying to make you into a chemist, but we do need to understand the basics of chemistry to understand how the body works. The chemists always like to tell me that physiology and anatomy is all applied chemistry, and they're probably correct, but we ignore them anyway. But no, we do need to understand chemistry, and particularly at the end here when we talk about DNA, our nucleic acids, and our proteins. Uh, the principles that we're going to learn here are going to be very important. Not only understanding how the body works and functions, but there's going to be a lot of applications for it as we go out throughout the semester. So I know this is maybe not necessarily everybody's favorite, but it is important for you to understand this, and you can understand it, which is important for you to understand. So, kind of like I was just saying on the introductory slide, really important to understand these basic principles of chemistry to understand the anatomy and more specifically your physiology of the human body. So, the picture on the right here, we've got some kidney function. The idea of acids and bases and buffers and the structural framework of all of our cells and our organelles. It all comes down to these principles of chemistry. Chemistry is the study of matter. And, so that begs the question, well, what the heck is matter? Well, matter is anything that takes up space and has mass. And I always find it interesting because then you go, well, what's mass? Well, the mass is the amount of material in matter. So matter is kind of like mass, and mass is kind of like matter. Anyway, we won't think too hard about it. The way we're going to look at it here, though, is mass is almost like weight. It's not weight, but just kind of for us to understand kind of what is going on here. So chemistry really is the study of the stuff that takes up space and has mass. Atoms are going to be the basic building block of chemistry, if you will. See, these are the smallest stable units of matter. There are going to be some smaller units, electrons, protons, and neutrons, called subatomic particles. But atoms are the smallest stable units, and so this is kind of what our focus is going to be on. Now, each atom is going to be made up of these subatomic particles, the electrons, the protons, and the neutrons. Electrons are going to be kind of the key as we talk about chemistry. It's the organization of electrons, the number of electrons, that dictate whether or not we get a chemical reaction. Electrons are very, very small, and they're going to contribute a little bit to the overall mass of an atom, but not much. So when we think of atomic mass, I don't like to think of electrons as being in there. Okay, protons and neutrons are going to make up the vast majority of the atomic mass. Electrons also have a negative charge to them, which is going to be important because atoms need to and want to be electrically neutral, generally speaking. And so the number of electrons we have is usually going to equal the number of protons. Protons are positively charged subatomic particles that are extremely large, and they are going to be found in the nucleus. The nucleus is made up of two subatomic particles. We have protons and neutrons. So neutrons and protons are found in the nucleus. Neutrons are also very large, but neutrons, as you can kind of guess from the name, they're neutral. They do not have an electrical charge associated with them. The atomic structure, well, basically what we're going to look at here is we're going to look at the periodic table of the elements, and we're going to be able to use these numbers to figure out the structure. We're going to be able to use these numbers and the symbol to figure out what we're looking at, how many protons something has, how many neutrons, how many electrons it has. Okay, so the first thing that we see on this periodic table is the chemical symbol. This is an abbreviation that is universally accepted. So even in areas of the world where they don't use the Latin alphabet, they will still use this chemical symbol here, the C for carbon. Everyone who works at all with chemistry knows the C stands for carbon. Some other examples that we have here: H, which is hydrogen; Cl, which is chlorine; O, oxygen; Hg is mercury. Sorry. These are usually going to come from the Latin names. This is one that always kind of confused me, is Na. I was like, well, it's sodium. How do they get the Na? Well, the Latin name for sodium is natrium, and so there's the Na that gives us our chemical symbol for sodium. We're just going to use some of the real common ones. So, like I said, I'm not trying to make y'all chemists. So we just want to make sure we understand that these are what the chemical symbol is and what they stand for. Now the top number here is going to be the atomic number. Atomic number. Now the atomic number is going to give us the number of protons in that atom. Now we can surmise, we can deduce from this knowledge the number of electrons, because remember, we want atoms to be electrically neutral. So protons have a positive charge. So for every positive charge, we need a negative charge. So if we have 6 protons, that means we have 6 electrons. But now if I ask you on an exam, what is the atomic number? What is the information we can get from the atomic number? What I want you to write down is the number of protons. Can we figure out the number of electrons? Yes, we can, but the atomic number is telling us the number of protons in the atom. The mass number is going to be equal to the number of protons and the number of neutrons. You'll recall the electrons, they do contribute a little bit to mass, but we're just going to kind of ignore that for right now. And so it's really easy to figure out the number of neutrons, because all you do is you take this mass number, 12, and you subtract the number of protons, 6. Okay, and so this leads us to the number of neutrons would be 6. All right, and this is I know it's a bad joke, but this is why people hate organic chemistry. Organic chemistry is all about carbon. If you look at it, how many electrons does carbon have? 6. How many protons does it have? 6. And how many neutrons does it have? 6. See, even here, right? This is the sign of the devil, 666. That's why organic chemistry is so stinking difficult. I know, bad joke, but you're laughing. Even if you're not laughing out loud, you're laughing inside. I understand. Isotopes are very interesting chemicals, if you will. These are so what we call an isotope of an element. So we look at like hydrogen, for instance. These isotopes of hydrogen, they all have identical chemical properties, but they have a different mass. So let's go back, and we just went over this, but what makes up the mass of an atom? The mass of an atom is basically made up, the majority of the mass is made up of the protons and neutrons. Now, we can't really change the amount of protons we have, because if we change the protons, we have to change the number of electrons, because remember, we need electrons and protons to be equal. So isotopes they have the same number of electrons, they have the same number of protons, but they differ in the number of neutrons they have. Okay, so example, I mentioned this hydrogen. Okay, hydrogen has 1 electron, 1 proton, and 0 neutrons, no neutrons. So that's what we call hydrogen 1. Hydrogen 2 is an isotope, okay, and it's got 1 electron, 1 proton, and now we add 1 neutron. So this hydrogen 2, this deuterium, is going to be quite a bit heavier than hydrogen 1, because now we've added a neutron. Okay, so the mass has increased, but the chemical properties are going to be the same. The chemical properties, I'm oversimplifying things here, but when we think of what makes an atom function, I want you to think of these electrons right here. So, so if the electrons stay the same, the number of electrons stay the same, then the chemical properties are going to roughly stay the same. So here we're just increasing the mass. Here, hydrogen 3, tritium, now we have two neutrons. Still the one electron, still the one proton, because they need to be equal. But now we've added two neutrons in there. Again, just increasing the mass of this isotope. Now, why do we care about isotopes? Because I know that's what you're asking already, right? We're going to make use of these isotopes quite a bit in healthcare. They are used for diagnostic purposes, and they are used for treatment procedures as well, treatment purposes. Basically, most of the time what we use are what are called radioisotopes. Radioisotopes are going to produce radiation. This radioactive isotope here is unstable, and it's going to break down. And as it breaks down, it releases the radiation in the form of either energy waves or particles. Okay, so this radioactive isotope wants to break down into a more stable isotope, if you will. Okay, and this radiation is what we use for a lot of our diagnostic and treatment procedures. Okay, great example would be well, here we have a PET scan. So what we do with a PET scan is we introduce a radioactive isotope. Sometimes you drink it, sometimes it's put into your blood, and this radioactive isotope travels around the body, and it tends to accumulate in areas that are most metabolically active. So, for example, a lot of times we'll use a radioactive glucose or glycogen. So it's going to go to these really metabolically active areas. And so if you see the scan to the left here, this is a PET scan. And down here, this is the bladder. And this makes sense because they're getting ready to pee out a big chunk of the radiation. The brain up here is bright red. It picked up a lot of this radioisotope, which again makes sense. The brain is very, very hungry, and so a lot of the glucose and whatnot it gets used by the brain. But this red spot right here in the middle of the lung field, that's abnormal. That's not okay. So what's happening here is that's most likely a tumor, probably a lung tumor from its position. And tumors tend to be very, very active. They tend to eat lots. They tend to grow very fast. And so they take up a lot of those radioisotopes. So a PET scan, put them in the machine, and the machine can read this radiation, which you can see just right there. Here are a few other examples of radioisotopes in use. So if we look down here, you see all these little white, almost looks like lice on the on this individual's X-ray. So this individual has prostate cancer, and what they do for prostate cancer is they inject these little tiny radioactive pellets into the prostate, and the radioactivity, the decay of these radioisotopes, destroys the tumor. And so they put hundreds of them in, if I remember correctly. So it's not the funnest of procedures, but it's certainly better than them going in and just taking the prostate out. So you can see these radioactive pellets for the treatment of prostate cancer. Here's a different type of scan of the thyroid, again using radioisotopes. Here would be a normal thyroid, and then you can see here's Graves' disease where we have hyperthyroidism. And then we talked about PET scan. Here again, diagnosis. Here's a normal PET scan, and here's somebody with Alzheimer's disease. And you can see the decrease in brain function when you compare the two. So that's why we use radioisotopes, and that's why I thought it was important that we at least understand the basics of what isotopes are and how they function. So energy levels. Atoms are electrically neutral, and we've mentioned this a couple of times already, but we'll mention it one more time. We need the same number of electrons and protons. Electrons have a negative charge. Protons have a positive charge, and so we need the same number there. Those protons and neutrons, they're found in the nucleus, and I'm going to say that they're relatively protected, they're relatively safe, but the electrons are not found in the nucleus. They are found floating around the nucleus in what the chemists like to call the electron cloud. They're just kind of floating around, kind of like the internet. It's just kind of there. I shouldn't. It's a little bit more organized than that. But the electrons are kind of on the outside, and this is important because these electrons are what are going to interact with other atoms and with other electrons. So we organize these electrons into what we call energy levels. I'm just going to use the term electron shells. And again, I'm not trying to make this a chemistry class, so I'm really making this as straightforward as possible, just so we can understand the human body. If you go take a chemistry class, they're going to get into quite a bit more detail with these energy levels and the electron shells. But what I'm talking about is just bare basics to get us through this class. As we increase the number of electron shells, as you can see here, like this would be the K shell would be the first one. That's got the lowest energy, and as we increase the number of shells, the outermost shell is going to have the highest energy. The outermost electron shell is known as the valence shell. And so this comes right out of your textbook here. The number of electrons in that valence shell determines the chemical properties of the element. Okay, so like I mentioned on the previous slide, the number of electrons and the electrons in that outermost shell. These are what interact with other atoms. So, for example, if you look at what we call our noble gases, if you look at your periodic table, it's on the right side there. Helium, neon, if I remember right. Okay, they are known as our noble gases. They are said to be inert. And what this means is they have a full valence shell, and because they have a full valence shell, they have no need to interact with other atoms. So this is why, for instance, you can inhale helium without it killing you. Okay, helium gas, because the helium gas does not interact with any of our body tissues. Now, I wouldn't recommend just sitting there breathing helium because you're going to run out of oxygen eventually. But that's kind of why you can breathe that helium in without, dying. If you tried the same thing with chlorine gas, and chlorine is right next to it's in the period right before that. Chlorine gas is highly reactive because it has an unfilled valence shell. Okay, if you inhale chlorine gas, you are going to die. But it all comes down to this valence shell. If it's unfilled, it's highly reactive. If it's filled, then it's inert. It doesn't react with other atoms or other cells or tissues in the body, for instance. This idea of valence shells leads directly to chemical bonding. So chemical bonds are going to be formed between two or more atoms that have unfilled valence shells. Okay, and I should have highlighted this one here. But bonds are formed when electrons are gained, lost, or shared to fill the valence shells. So the whole point of these atomic interactions is to fill the valence shells. Atoms are not happy until they have a full valence shell, and we will talk. Give me just a minute here, and we'll talk about what these valence shells look like and how we fill them, and what makes them be full, if you will. All right. So the first type of bond that we want to talk about is an ionic bond. Ions are atoms that have an electrical charge. Now, this is one of those things that I just completely missed and for like years of my schooling, I had no idea what we were talking about with these ions or electrolytes. Electrolytes are basically just ions. So ions are atoms that have an electrical charge. It's in red. Please make sure you know that. These ions are going to be formed when an atom either gains an electron. If they gain an electron, they're going to be negative because electrons have a negative charge, or lose an electron, in which case they have more protons than they have electrons, so they'll have a positive charge. So an electron donor, somebody who gives up an electron, so they're going to be positively charged, and they are known as cations. Positively charged ion is going to be a cation. An electron acceptor, okay, one of these atoms that receives one of these donated electrons, is going to have a negative charge and is called an anion. And this is the key right here to ionic bonds, okay? Cations, positive charge; anions, negative charge. The positive and the negative charges now attract each other, and that's what's going to form the bond. I want you to think of this bond kind of like a magnet. You put the positive side of one magnet and the negative side of another magnet, and what do? They stick together. Okay, so let's see how this works. So let's see if I can draw this out. Certainly not very good at drawing on a computer with a mouse here. But let's look at hydrogen first. Okay, so hydrogen has you'll recall it has one electron and one proton. Okay, so with that one electron, the first thing we want to do is we want to draw a shell. So this is the innermost shell, and this innermost shell can have a maximum of 2 electrons. Okay, now hydrogen only has 1, so let's put 1 electron right here. Okay, so this is hydrogen's valence shell, and hydrogen does not have a full valence shell. Okay, because hydrogen is missing an electron. Okay, because remember, this innermost shell can have a maximum of 2 electrons. So hydrogen is one of those that's going to be highly reactive. Okay, now if we look at its next door neighbor, helium, okay, helium has 2 electrons. Okay, so we draw this valence shell in there. It's supposed to be round, but that's okay. And now we can draw in the 2 electrons. Okay, and now helium, because this is the outermost shell, but because it's the innermost shell as well, it can have a maximum of 2 electrons. So helium has a full valence shell. And remember, we're gonna term these atoms with full valence shells as inert. So helium is not going to be very reactive because it doesn't need an electron, and it certainly doesn't want to give up an electron because otherwise it would not have a full valence shell. So helium is just fine over here by itself. Now, 2 of the ones that are going to form ions that we need to talk about are going to be sodium and chlorine. Okay, so sodium, hold on here, write that. Sodium is going to have 11 electrons and 11 protons. Okay, so we draw that first shell, and how many electrons go in this first shell? 2. Okay, and so we still have 9 more to go, so we draw a second shell. Now the second shell can have a maximum, for our purposes, the rest of the shell, so the first shell can only have 2 electrons, but all the rest of the shells can have a maximum of 8. Okay, so we still have 9 more, so let's go, we'll put 1 there, 1 there, one there, one, oh, I'm already failing here. Okay, so that should be 8 plus the 2 is 10, which means we have one more electron to add. So we add another shell, and we'll put our little electron right here. Okay, so this outermost shell now is the valence shell, and that electron right here is called the valence electron. So you notice sodium is incredibly unstable because how many electrons does it have in its outermost shell? It's got 1. How many does it want in that outermost shell, that valence shell? It wants 8. And so sodium is desperately going to try to figure this out. Now right next to it is going to be the buddy chlorine. And chlorine has 17. Actually, let me change pen colors here. Okay, so let's do it here. All right, so first shell, we have 2. Okay, then we draw the next shell. And how many can we fit in the next shell? We can fit 8. 2, 3, 4. Maybe I'll just scribble. 5, 6, 7, and 8. Okay, so that should give us a total of 10. So we need 7 more. So we draw this next shell. And 1, 2, 3, 4, 5, 6, 7. Okay, so can you see the problem with chlorine? So chlorine in its outermost shell has 7 valence electrons. How many does it want? It wants 8. Okay, so when we look at these two next to each other, what do you think? Do you think it's easier for sodium to gain 7 electrons to fill its valence shell, or just to lose this 1 electron? Probably lose that electron. And on the flip side with chlorine, do you think it's easier for chlorine to gain 1 electron or to lose 7 electrons? It's going to gain 1 electron. And so this is how sodium and chlorine are going to be best buds. Because what's going to happen here is sodium's going to say, hey, I have this extra electron, do you want it? Okay, and it's going to donate this electron to chlorine, and chlorine's going to take it up. So there's that donated electron. So now chlorine has a full valence shell, and because sodium lost this 1 electron, we go to the next lowest shell, which is now its valence shell, and this is now full because it still has 8 electrons, and so sodium is happy and chlorine is happy. Okay, but now because we've lost and gained electrons, we are now going to become ions. Okay, so sodium normally has 11 electrons and 11 protons. Okay, but now in this case, what happened? What did we do? We lost an electron, and now we only have 10. Okay, so now you can see we have more protons, 11 protons to 10 electrons. So this atom now has a positive charge because we have 1 more proton than we have electron. Okay, now chlorine, do the same thing over here. Chlorine usually has 17 electrons and 17 protons. Okay, but because of that extra electron it got from sodium, it now has 18. Okay, and now you notice we have more negatively charged electrons than we have positively charged protons. So chlorine is now going to become an anion, and it's now charged. Okay, and because of these two opposite charges, right? Opposites attract. Okay, so you can see here the attraction of these magnets to the big magnet. Chlorine and sodium are going to form an ionic bond. Okay, they are now going to stick together just like magnets. Okay. Now, just as I'm looking over here at the picture, what do like charges do? They repel each other. Now, these forces here are going to be extremely important because they're going to explain the movement of ions in the body. And this is going to be very important. It's going to be a recurring theme that we're going to talk about over and over and over again. Opposite charges attract, like charges repel. So here, sodium and chlorine, because they are ions, oppositely charged ions, they're going to be attracted to each other, and they form this ionic bond. And we now have a molecule called sodium chloride, more commonly just known as table salt. Okay, so this is ionic bonding.
We're now talking about our next type of bond, which is going to be covalent bonds. Now, covalent bonds are going to be they're going to be different from our ionic bonds. So remember, ionic bonds, we have atoms that donate or accept electrons, and they become charged. They become charged ions, and then those oppositely charged ions are going to be attracted to each other. With covalent bonding, the electrons are not going to be given and received. Instead, they are going to be shared. And by sharing these electrons, each atom will complete the valence shell. A great example is, I'm not going to draw these, we'll just kind of look at them here in the pictures, would be hydrogen. So hydrogen is very, very rarely seen just by itself. So you don't see a single hydrogen atom. But hydrogen gas, for instance, has two hydrogen. It's H2. And what happens here, as you can see, we have two atoms of hydrogen, each one with an electron. And what they do, instead of saying, "Hey, give me your electron," and here, have my electron, they say, "How about we share these two electrons?" So now, because they have two electrons, this atom has two electrons in the valence shell, which fills the shell. And this atom has two electrons that fill the valence shell. So they are going to be sharing these electrons. Here you can see the same thing with oxygen. This will be a double bond. Instead of sharing just one pair of atoms, they are going to share two pairs of atoms. And triple bonds, so like nitrogen gas, this one down here, they actually share three pairs of electrons. Okay, but no one's taking, no one's giving. They're all just sharing, and so that's what's going on here. Look at all these dogs sharing the stick. Except for the little runt over here, he doesn't get any part of it. But generally, they're pretty good sharers. Now, with these covalent bonds, we do have to get a little bit more specific. We have to talk about polar and non-polar covalent bonds. With a covalent bond, everything is going to be electrically neutral. Remember, if we're giving or receiving, we're creating ions. We're not creating ions with covalent bonds. But just because everything is electrically neutral, meaning we have the same number of electrons as we have protons, does not mean there's not some electrical charge to it. And let me explain that a little bit more. Okay, so here would be a molecule of water. Okay, we have two molecules of hydrogen and one molecule of oxygen. Now, oxygen here, and I'm going to try to describe it this way, is extremely massive. It's got a mass number of about 16, where hydrogen has a mass number of 1. So even with both these hydrogens, they have a mass number of 2 compared to this gigantic, massive, relatively speaking, oxygen atom. Now, these three atoms here are going to form covalent bonds, where oxygen is going to share a pair of electrons with this hydrogen, and it's also going to share a pair of electrons with this hydrogen. But it's not going to be equal sharing. Okay, the way I always kind of think of this is like you've got three best buddies on summer vacation. Okay, and they're just going to kind of bounce around each other's houses. You know, they just like best buddies do. But one kid has a swimming pool and a ping pong table. None of the other two have a swimming pool or a ping pong table. So at any given point, where are you most likely to find these three kids? Probably at the swimming pool house. Okay, now, I mean, chances are they could be at the other two houses, but most likely you're going to find them at that one house. So oxygen, because it's so massive, is going to attract those electrons more than these two atoms of hydrogen. So at any given time, those two shared electrons from the hydrogen atoms are more likely to be found around the oxygen nucleus. Okay, which means this hydrogen at any given point most likely does not have any electrons, and neither does this one, whereas those two extra electrons are most likely going to be in oxygen's sphere of influence, if you will. Now, if you just do the count, if you count it all up, it is going to be electrically neutral. There's going to be the same number of protons as we have electrons. But because of this unequal sharing, okay, you can see this symbol right up here. This means that this oxygen atom, as part of this molecule, is going to be slightly negative, and the two hydrogens are going to be slightly positive. Now, this becomes extremely important, the polarity of these bonds, because this is going to allow us to make hydrogen bonds. And so this is what's known as a polar bond. We now have a negative pole and a positive pole because of the unequal sharing of electrons. Now, a non-polar bond is where we get an equal sharing of electrons. If we were just to stick these two hydrogen atoms together and make hydrogen gas, okay, there's no inequality in the sharing of the electrons. So that shared electron at any point is going to be found around both of them. So it's not an unequal sharing. These two friends, they both have the same, Xbox games. I mean, there's really no difference in their home. So it doesn't really matter. Your chances of you finding them in either home is about the same. Okay, so that would be a nonpolar where we don't get the slight positive and the slight negative charge. Now that polarity is going to lead us to our third type of binding that we're going to talk about here, bonding that we're going to talk about, which are going to be hydrogen bonds. Hydrogen bonds are extremely, extremely important, but very, very weak and transient, meaning they come and they go very, very quickly. And this is going to be bonds that are formed when the should be a slightly positive charge of hydrogen. I got that flipped out. The slightly positive charge of hydrogen attracts a slightly negative charge of another molecule. So I'll go ahead and flip those in your notes there. I need to make that correction. Okay. So here we are. Here's our water molecule. You can see the oxygen in red and the hydrogens in blue. So this hydrogen right here is slightly positive, and this oxygen right here is slightly negative. Now you recall from when we talked about ionic bonds, opposite charges attract. So the slightly positive charge is attracted to the slight negative charge of this oxygen, and so we form this very weak and very temporary bond between these two molecules of water. Okay. Now, why are they weak and why are they so transient? Well, because as soon as that electron from here finds its way back to this hydrogen atom, then this slightly positive charge goes away and this bond will break. Okay, but then that electron goes back into the oxygen, and the slight positive charge is there again, and we can reform this bond. So these bonds between all these water molecules are constantly breaking and reforming and breaking and reforming, which explains many of the really important physiological properties of water, which we'll talk about here on the next slide. But these hydrogen bonds are extremely important when it comes to life. Okay, not only is it important when it comes to water, but hydrogen bonds are going to, for example, keep our DNA together. So we'll see hydrogen bonds help keep proteins together. They're very important bonds that we'll talk about a few different times, particularly in this lecture. So what are the properties of water that make it so useful and so important to life? First of all, water is going to be the universal solvent. Okay, so when we look at a solution is made up of the solvent, in this case water, and then the solute. And so if you think of your blood being a big solution, the major solvent in that solution is going to be water. If you think of the cytosol, the fluid portion, or let's just go cytoplasm of your cells, inside of your cells, the fluid portion, I mean, that's just, again, one basic big solution. The fluid portion, water is the solvent, and everything that's inside of our cells is going to be dissolved. It's going to be a solute in that solution. Most chemical reactions will need to take place in water, and so reactivity is an important function of water. That's why we have so much water. But 60 to 70% of our body is made of water, and a good reason for that is the need for these chemical reactions to take place in water. Lubricant. Very little friction between water molecules. And so in areas of the body, like inside of our joints where we have synovial fluid, or in our serous membranes, for example, around the heart, the lungs, and the abdominal viscera, we have serous membranes that produce a serous fluid, which allows us to, for example, with our lungs, breathe in and breathe out without your lungs rubbing up against your chest wall, which is very, very painful when it does happen. So water is a fantastic lubricant. High heat capacity. This is actually kind of interesting. I'm kind of nerding out on it a little bit more every time I learn a little bit more about it. It makes a bigger impression on me. But when we talk about heat capacity, it's defined right here. It's the amount of heat required to raise the temperature of a substance 1 degree Celsius. And water has an extremely high heat capacity, which means we have to put a lot of heat into water to raise the temperature. And this comes down to those hydrogen bonds that we talked about on the previous slide. Okay, this is also what's known as thermal inertia, meaning it takes a lot for water to go one direction or the other. Water is very, very stable. This is very important because this is going to allow us to, for example, come back to these chemical reactions. Our water temperature inside of our body is not going to change very quickly. It takes a lot of heat, a lot of energy to change that water temperature, which means it's a relatively stable environment for all these chemical reactions to take place in. Okay, it's like I said, just a very, very high heat capacity, which I think is really cool, particularly when you start looking at these chemical reactions. Kind of the other aspect of this heat capacity we want to look at here is that water also carries heat with it, and so when we sweat and we put water on the surface of our skin, as that water evaporates, as it turns into a gas, it's going to take a lot of that heat with it. The final couple of properties of water that I want to mention here are principles of adhesion and cohesion. Okay, so adhesion is when water sticks to other things. So, we live in Ohio, it rains quite a bit here. When we think of rain on a windshield, that water is sticking or adhering to your windshield. So that's one of the important principles of water. Cohesion is very similar, but what it means is water sticks together. I remember I think it was probably middle school, we were doing an experiment where we took a little dropper and a penny, and we put water drops one at a time on top of this penny. And pretty soon, the water sitting on top of the penny was actually thicker than the penny itself. And it actually formed this little bubble because all these water molecules clung together. And again, that comes back to the hydrogen bonds that are forming between the water molecules. Now, when we talk about chemical reactions, a chemical reaction is when a bond is either created or destroyed between atoms. And cells in the body as a whole are going to use chemical reactions to function. So we'll talk about this a little bit more in depth in a little bit later in this lecture as well as in the next lecture. But these chemical reactions, this is basically what makes the human body function. This is basically what makes life function. As soon as these chemical reactions stop, life stops. So we do have this term here called metabolism. Metabolism is, I don't want to say it's misused, even though that's what I usually say. There are a couple of definitions of it. And most of the time when we think of metabolism, we think of just how fast we process food, which is partially true. But I hear all the time, oh, they've got a fast metabolism, or they have a slow metabolism, which I understand. But metabolism, the strict definition here, is the sum total of all the chemical reactions that take place within the body. So we are constantly undergoing chemical reactions. Sometimes we're building, sometimes we're breaking down, but there's just there are constant chemical reactions taking place within the body. And metabolism is the sum total of all of those reactions. If we're looking very generically, individuals that gain weight have more anabolic reactions, meaning they have more building reactions. The people that are losing weight have more catabolic reactions, which we'll talk about here, but those are the ones that break down. A couple other just vocabulary words here as we talk, the reactants and the products. So the reactants are the atoms or molecules that are going to be undergoing the chemical reaction, and the products are what happens after the bonds are either created or broken. The first type of chemical reaction we're going to talk about here is what's called a decomposition reaction. Okay, now I'm going to use these terms kind of interchangeably: decomposition and hydrolysis reaction. Okay, so I don't think there's much of a difference there. And then we're also going to use this term catabolism. And basically what we have here is here's our product AB, and like you can kind of gather from the name decomposition, we're going to break the bonds or the bond between these two products or reactants and come up with product A plus product B. Now, usually what we have to pull out of the bond between these two reactants here is going to be water. Okay, so here we are. We have, and these are good pictures, but unfortunately this one's going backwards. So what we're going to do, sorry, we're not pulling water out, we're putting water in. So we start here with this big, long, multiple subunits. And what we do is we add water, H2O, we put it in. Okay, hydrolysis, we're going to cut that water, and half of it's going to go on this subunit and half is going to go on this subunit, and we've now broken the bond between these two subunits here. Okay, so hydrolysis, we break water, and water is going to break apart the bonds between these two molecules. Breaking apart, the collective term we're going to use for this is catabolism. So a catabolic process or catabolism are all the decomposition reactions that take place in the body. A synthesis reaction, on the other hand, well, it's quite the opposite. To synthesize means to make. This is also known as a dehydration reaction. And we're just going the opposite direction. So we have reactants A and B, and we are going to put the two together to come up with product AB. And here, so with our decomposition reactions, we added water to break the bond. Here we're going to pull water out to allow these two reactants to come together. So here we are. Here's reactant A, and here's reactant B. And you can see what we're going to do is we're going to pull out OH, an OH group, and hydrogen. We're going to pull the water out, which is why this is also known as a dehydration reaction. In some textbooks, you may see condensation. I'm not going to use that term because I just want you to kind of focus on the water here. So dehydration. We pull the water out, and now you can kind of think of it as what's left are two sticky ends. And so this subunit is going to stick to this subunit, and we're going to form a bond between the two. Okay, now this is also known as anabolism, which is just a collective term for all the synthesis reactions that occur in the body. So if we're kind of keeping track, well, let me go to the next slide and we'll kind of sum this all up. So here is metabolism. If we look at this whole picture right here, metabolism is the sum total of dehydration or synthesis reactions, anabolism, and hydrolysis or decomposition reactions, also known as catabolism. So metabolism is the sum total of anabolism and catabolism that takes place in the body. And it's really impressive when you start digging through all the biochemistry, because we start off, like eating, what do we want to eat here? A piece of bread. A piece of bread is basically pure sugar. So let's look at it that way. So we have this long carbohydrate chain from our piece of bread, but this long carbohydrate chain can't get into our cells, and we can't absorb it into the intestinal wall. So what do we have to do with that long chain of carbohydrates? We have to break it down. And we're going to use things like salivary amylase, a little bit in the stomach. We also have pancreatic amylase in the intestines. And we're going to break all these down into smaller and smaller subunits, or into smaller and smaller units that we can then we can actually absorb through the intestinal lining. We can put it into the blood. And then these small units, most of which are going to be glucose, will travel around until they enter another cell, and then from there we build them back up. So here's glucose and here's glucose, and we put them together to make maltose, for instance. And we're going to use these long carbohydrate chains to make things like on the cell surface we have things like glycoproteins, which are chains of sugar with a protein on the end. The glycocalyx. We use these carbohydrates for other things. So we take it from a form the cell can't really use. We break it down into these small subunits, and then the subunits go into the cell, and then the cell builds it back up into whatever it needs to function properly, which is really, really awesome if you kind of think about this for a second. Taking something that we can't use, we break it down into the individual subunits, and then we use those subunits to build it back up into something we can use, which is just absolutely fascinating. Okay, but this is kind of how these anabolic and catabolic reactions work. Just to kind of give you a hint, because it's sometimes it's hard to keep track of all these: anabolism, catabolism, metabolism. So I always remember just anabolism. And whenever I think of anabolism, I think of anabolic steroids. And what's the point of taking steroids? Well, it's to build muscle. So anabolic reactions, we're building things, and catabolic reactions, we're breaking down. The last type of chemical reaction I want to mention here are the exchange reactions. And I always say these are the swingers of the chemical world. So we're just exchanging. We still have the same number of chemical bonds, but we've broken some and then recreated them a little bit differently here. So we have reactants AB and reactants CD. And all we're going to do is we're just going to exchange these. Okay, so AD is the new product and CB is another new product. So if we have just one bond between AB and one bond between CD, we We still have the same number of bonds. It's just everything's reorganized a little bit differently. Okay. Now all these reactions, these exchange reactions, the dehydration, the hydrolysis reactions, they are all theoretically reversible, meaning going back and forth. There are a lot of things that kind of dictate if these chemical reactions occur and how they occur and if they're reversible and whatnot. So we're not going to get into that. I think that's more of the domain of a chemistry class. But just understand that these chemical reactions are, the arrow is not always going this way. They can certainly go back if the conditions are correct. Now we look at the sum total of chemical reactions. There are two big problems that we have to overcome before the body can function properly. The first one is that these chemical reactions, if left to their own devices, are going to occur too slowly to be of use by the body. Okay, like if we, if we were to take in some carbohydrates, for instance, and we were just wait for it to magically break apart and then magically come back together in the form of glycogen, for instance, that's not going to happen. That's going to be too slow for anything to happen that's compatible with life. Okay, so somehow we need to be able to speed up these chemical reactions. The second problem that has to be overcome is the amount of energy that is required for some of these chemical reactions to occur. Really, one of the ways that we get chemical reactions to occur is we increase the heat. The problem is that the temperature, the heat, the energy that is required for a lot of the chemical reactions to occur in the body really, again, aren't compatible with life. We'd have to be several hundred degrees Fahrenheit or even several hundred degrees Celsius in some cases to achieve the appropriate amount of energy for these chemical reactions to occur. So these are two major problems. Okay, chemical reactions, if we were wait for it to happen randomly, are too slow, and they require too much energy or heat to be compatible with life. The answer to both of these problems are enzymes. Now, I remember as a student sitting there going like, man, why do I care about these enzymes? It doesn't make any sense. And now I can't get enough about enzymes. I absolutely love talking about enzymes. I think they're absolutely amazing and just incredible tools that our body uses. And the variety of enzymes that we use and the variety of chemical reactions that they help out it's just incredible. It's just incredible. Okay, but enzymes, they're going to help solve these problems for us. So the first part of it here is enzymes are biological catalysts. Okay, and a catalyst is anything that speeds up a reaction without being changed or used up in the reaction. Okay, so they are just facilitators. They're going to make sure that these chemical reactions occur without being involved in the reaction otherwise themselves. Okay, so they're not changed by the reaction. So by being a catalyst, what they do is, one of the big things they do is they lower what they call the energy of activation. So instead of needing the energy to be like several hundred degrees Fahrenheit, now these chemical reactions will be able to take place within a range of temperature that is compatible with life. Okay, and amazingly, and when we say it makes complete sense, but most chemical reactions will occur we're using enzymes. Most enzymes will function right around body temperature, 98.6. That is the ideal temperature for these enzymes to work at. Okay, but these enzymes are going to take all these chemical reactions that we need to do, and they're going to lower the energy that it takes to do them. Okay, now the other thing that enzymes do is kind of think of them like a matchmaker. Like they're going to bring these reactants together so that the chemical reactions can occur. So now these chemical reactions will go just about as fast as not just about, they will go just as fast as we need them to go to be compatible with life. All of this because of enzymes. Now I'd like to show this picture over here. One, so you'll feel sorry for me. I took a biochemistry class in my chiropractic studies where we had, so this right here is what's known as the Krebs cycle or the citric acid cycle. We had to memorize each one of these steps. So pyruvic acid to acetyl-CoA, acetyl-CoA to citrate, citrate to sisaconitate, sisaconitate to isocitrate, and so on. It's a cycle, it goes all the way back. Okay. Not only did we have to memorize every step, but we also had to memorize all of the enzymes that catalyzed each step. Okay, so to be able to go from citrate to sisaconitate, we need this enzyme right here, aconitase. Now, most enzymes are going to end in the word or in the with the ending ase, so that's usually what we're looking for an enzyme. So, but we can see how important. So here's an enzyme, and this one happens to catalyze two reactions. But every other step along the way, look at all the different enzymes we have to use just for this one part of our metabolism. It's a relatively small part. So enzymes are all over the place and extremely important. Okay, so if we went over here and let's say we had a genetic problem where we couldn't make aconitase, body can't make the enzyme. Then there's no way for us to go from citrate to sisaconitate. And this whole metabolic process that we're looking right here, this pathway stops right here. Okay. And just to be overly dramatic, you would die because you could no longer produce ATP. This is part of the cycle that helps us produce ATP, enough ATP to live. So without that enzyme, this whole metabolic pathway stops, and that's it. We're done. We're no longer alive. Would eventually citrate turn into sisaconitate? Absolutely. Would that be in a timely enough manner that we don't die? No, it wouldn't. Okay, so enzymes I think are just the coolest things. They're so cool, in fact, that I have a second slide just to look at the pictures here. So here we are. Here are what we call our substrates. You can kind of think of these as the reactants. And here's the enzyme. So you can see how the substrates, they have a very specific shape to them. Enzymes are highly specific. Okay? Because you can see that in the enzyme, what we call the active site matches up with the shape of the two substrates. You see how they fit in there nice and neat? They call this the lock and key model, if you will. Okay, so these two substrates are going to snuggle right up inside of this enzyme, and the enzyme is going to lower the activation energy so that bonds can form between these two substrates. And as soon as that's done, off they go. We now have a product. You can think of this, well, it's a product, okay? And then you notice the enzyme is unchanged and is now ready to accept more substrates, okay? So enzymes, the concentration of enzymes many times will dictate the speed of chemical reactions. Now in this one here on the left, we are building, but we can certainly go the other direction. Sometimes these enzymes will catalyze decomposition reactions. So here's our substrate, okay? And again, it's going to fit in, lock and key model. It's going to fit into this enzyme. And instead of building a bond, here this enzyme is going to break a bond, and out comes our products. So we'll see this, I mean, both of these frequently when we're breaking down carbohydrates, for instance, okay? We have a long chain of carbohydrates fit in those enzymes, and the enzymes will start breaking bonds between these two molecules of carbohydrates. So we take this long chain of carbohydrates, let's say a starch. We just had a nice potato, very starchy, so a lot of starch. We can't use starch in our body, and so one of the things we need to do is we need to break it down. So we're going to take that starch and we're going to break it down into its individual glucose molecules. Then we can take those glucose molecules and we can come over here and we can start building things with other enzymes. So really, really cool. I kind of like this little GIF here on the side. You can see the same thing. So here comes our substrate. It fits in there nice and neat. There's our chemical reaction, and we break the bond, and out come the products. So pretty cool. Acids and bases are, I want to say they're one of the more confusing things, at least for me, one of the more confusing things. They're very, very simple and straightforward, but at the same time, it doesn't always kind of click with me. But let's talk our way through acids and bases here and see what we come up with. The first thing we need to understand with acids and bases is when molecules are introduced to water, and even water itself, but when molecules are introduced to water, they break up. This is a process we call dissociation. And so we're going to use this term as we describe acids and bases as dissociation or breaking up, the breaking of molecules. An acid is any substance that dissociates in water. So when you stick it in water, it breaks up. And when it breaks up, it releases hydrogen ions, also known as protons. Okay, so here's an example for you. HCl, this is hydrochloric acid. If you were to dump HCl into a beaker of water, then the bond between the hydrogen and the chlorine atoms would be broken, and we would release hydrogen ions and chloride ions. Okay, and so this happens to be a very, very strong acid, because in water it almost completely dissociates. So when you stick hydrochloric acid into water, almost all the hydrogen ions separate from the chloride ions. Okay, so that's what an acid is anything that when you stick it in water, it breaks up and it releases hydrogen ions. Now just kind of keep that in mind. When we go to our next slide, we're going to talk about the pH scale. And the pH scale is all about the hydrogen ion concentration. Now on the flip side of that, a base, and I'm going to look at it two ways here, is any substance that takes up free hydrogen ions, so anything that gets rid of these hydrogen ions that are produced from the acids, or that releases what we call the hydroxide ion. Now again, this is kind of two ways of looking at the same thing, because this hydroxide ion is you can see it's got this negative charge. It's going to be attracted to this positive charge, so it's going to take up those free hydrogen ions. But bases don't have to just have the hydroxide ion. So here we have sodium hydroxide. When we stick that in water, it dissociates, it breaks up, and we get this sodium ion and we get the hydroxide ion. Very similar to our acid example above, the hydrochloric acid. This it dissociates almost completely, so is a very, very strong base, meaning if we use sodium hydroxide, we get lots and lots of these hydroxide ions. Now what happens when we put a base in with an acid? Okay, well here, let's look at it. Here's our hydrochloric acid, and we add sodium hydroxide. And what is going to be produced, so remember these are our reactants, these are our products, okay? We're going to have a salt, sodium chloride, just table salt, and water. So when an acid and base combine, we're going to get some type of salt, and we are going to get water. The pH scale is going to be how we measure how acidic or basic a solution is. And the easiest way to kind of think of this is going to be the hydrogen ion concentration. So these little brackets around hydrogen ion, that just means concentration. So the hydrogen ion concentration has an inverse relationship with pH, meaning as hydrogen ion concentrations increase, pH decreases. Inverse means it's flipped. It's going the opposite direction. So as hydrogen ion concentrations increase, pH decreases. Now, what is going to cause an increase in the hydrogen ion concentration? acids. So the more acidic something is, the lower the pH, because the more acidic it is, the more hydrogen ions that it releases, and the more hydrogen ions, the lower the pH. Okay. Now, on the flip side of that, if something is going to decrease the hydrogen ion concentration, a base, that means the hydrogen ion concentration is going to decrease, which means the pH is going to increase. Okay. 7 is going to be neutral on our pH scale, and there is this is not just random. So this happens to correspond with just pure water. So pure water will dissociate into hydrogen ions and hydroxide ions. So it ends up being completely neutral. Okay, and so that's where we get this neutral on the pH scale is pure water. So anything less than 7 is considered acidic. Anything greater than 7 is considered basic or alkaline. Now, if we look at this in regards to the body, a great example would be pH of our blood. So the pH of our blood is considered to be 7.4. Now, with all things that's an average, there's going to be a range here, and we usually range between the 7.35 and 7.45 is what we consider to be normal pH of the body. Where we run into issues is when we're below 7.35 or we get over 7.45. So if anything is over 7.45, this is considered now to be alkalosis. Anything below 7.35 is now considered to be acidosis. Now, the only thing that's kind of tricky here is, even though you're looking at this and saying, well, we're still over 7, so why is it acidic? Well, because relative to neutral of the body, the pH of the body, this is more acidic than we're used to. So this is all relative to the pH of blood, which is 7.4. So anything below 7.35 is considered acidosis. So here are a few examples. I always kind of like looking at these. So some things that are really acidic. So if we flip over on this side, things like soda, beer, vinegar. The other name for vinegar, the chemical name for vinegar is acetic acid. Okay. There's stomach acid and lemon juice way down there. On the flip side over here, we're going to have things like ammonia, oven cleaner. I don't know anyone who uses oven cleaner, but I always use that as an example on these things. Baking soda, okay, those so that you can see a lot of things over here on the basic side. Much of what we eat is actually over here on the acidic side, which is kind of, I don't know, kind of interesting to think about, I guess. Now the body has designed itself to deal with big changes in acids and bases. Okay, and what we have in the body we have what are called buffers. So let's say, like on a stupid dare, you take you go, I'm going to drink a whole cup of lemon juice. Okay, just straight lemon juice, which is extremely acidic. So let's say you drink this cup of lemon juice. Does that mean that the pH in your body is going to drop, and now everything's going to melt in the acid that is the lemon juice? No. We have buffers that are going to prevent large changes in the pH inside of our body. Okay, so some of the buffers are going to take up extra hydrogen ions. Others are going to take up these hydroxide ions. Okay, so if we look right here, we have excessive amount of hydrogen ions. Here's a real common buffer we use, bicarbonate. And so this H plus, the HCO3 negative is going to combine. So this bicarbonate is going to take up the excessive hydrogen ions, and it's going to form carbonic acid. Now I do like to highlight this here. We're still forming an acid. Okay, if we drink a whole bunch of lemon juice, is our pH going to get lower? Absolutely. Is it going to get low enough? Is it going to lower down to the lemon juice acidity? No, it's not. So this carbonic acid here is what we consider to be a very, very weak acid. So the pH is going to lower a little bit because we are creating an acid, but it's not going to swing as wildly as it would otherwise. Okay, now here we have an excessive amount of hydroxide ions. Okay, along comes our acid. Here's our carbonic acid again, and now it's going to release bicarbonate, as well as water. Because remember we talked acids and bases. When we put the two together, what do we end up with? We end up with the salt and water. Okay, so if we look at this, let's look at this in regards to carbon dioxide. So we're constantly producing carbon dioxide in the body, and carbon dioxide is actually considered to be more acidic. Okay, so carbon dioxide will combine with water to form our carbonic acid. Okay, and so if we have an excessive amount of CO2 in the body, we start to become acidic because we make lots and lots of this carbonic acid. Now this carbonic acid can dissociate into carbonate and hydrogen ions. Okay, so this right here is a buffering system where we take this what would be considered a really, really acidic substance like CO2, okay, because it forms this carbonic acid, and we make it into a weaker and weaker acid so that it's still going to get changed, but it's not going to be an excessive amount of change. The way I always kind of describe this in class is kind of like when you go, let's say you go to Thanksgiving, right? And everyone's got that crazy Uncle Frank who likes to talk about politics, okay? And if you're sitting right next to Uncle Frank, you're going to get this big blast of political nonsense. I'm sure you don't like whatever he has to say, okay? And so you don't want to sit right next to him because that's gonna make your meal absolutely miserable. So what do you need? You need buffers. So you need to put mom, dad, grandma, Aunt Eileen all in between you and Uncle Frank. Now, are you still going to get some of his political diatribe? Absolutely. But are you going to get as much of it as if you were sitting right next to him? No. So that's kind of the ideas behind these buffers. We introduce something really acidic or something really basic, and these buffers are going to take that and they're going to change it into weak acids or weak bases. There is going to be a change in the pH, but it's not going to be as dramatic or as large as it would be without the buffer system. Now we need to talk about organic versus inorganic compounds. We're going to spend the rest of this lecture here talking about our organic compounds, but we're just going to at least introduce inorganic compounds here. Inorganic compounds, generally speaking, we're going to say do not contain carbon or hydrogen as their backbone. When we look at these organic compounds, they are made up primarily of carbon and hydrogen. We sometimes wrongly think, inorganic don't contain any carbon, and that's not true because carbon dioxide has carbon in it, but is considered to be inorganic because it doesn't have carbon and hydrogen as the backbone there. Okay, some other inorganic compounds we're familiar with: oxygen gas, O2, water, H2O, lots of salt, sodium chloride, and then the acids and bases we were just talking about. So organic compounds, on the other hand, carbon and hydrogen are going to be the backbone. If you take OChem, and most of you are going to have to take OChem at some point, you're going to be introduced to these molecules, these bigger molecules that have really, really long carbon chains. You'll see some fatty acids that have 22, 24 carbons in length. And most of those carbons are going to be attached to hydrogens. We'll see a few examples of carbohydrates and fats here in a moment. Carbon and hydrogen is going to be the backbone. Now, very important here, I want you to understand this part right here, okay? So these are going to form our macromolecules that we're going to use to build the body. Now, macro means big molecules. So big molecules are usually made up of smaller parts. These smaller parts are known as monomers. Okay, monomers are going to be the building blocks of our large organic compounds. So monomers come together to form polymers. So for almost all of these, a few of them we don't have real clean ones, but for almost all of these, I'm going to ask you what are the monomers and what are the polymers, okay, of each of these large organic compounds. So the big organic compounds we have are big macromolecules, carbohydrates, lipids, proteins, and nucleic acids. So I just kind of mentioned all this on the previous slide, but here are the big macromolecules. Monomers, these are going to be the building blocks. These are going to be the smallest units of these macromolecules. We put a bunch of these monomers together and we form a polymer. Okay. This is not a chemistry class, and so I guess I just at least wanted to mention this, but we're not really going to get into it. And please don't memorize any of these. None of these will be on the on a test I give you. But there are going to be, for example, when we look at the amino acids, the building blocks of proteins, the basic building block of all of our proteins are roughly all the same. Okay, the big difference on each one of these amino acids is going to be what we refer to as the functional group. So the functional group is going to be a group hanging off of the basic framework that is going to react with other molecules. So whether or not these molecules interact usually comes down to their functional groups. And you can kind of see just from the name, functional, they're doing something. Let's start our conversation here. We're talking about carbohydrates. So when we look at carbohydrates, and it's not a pure definition here, but generally speaking, carbohydrates are going to contain a ratio of 1 carbon to 2 hydrogens to 1 oxygen. Okay, so when we look down here at a few examples of our monosaccharides, we'll get to that here in a second. You notice C6H12, so we have twice as many hydrogens as we have carbons, and then we have the same number of oxygens as we have carbons, so C6H12O6. So this is the basic setup here of our carbohydrates. 1 to 2 to 1 ratio, carbon, hydrogen, and oxygen. Carbohydrates are going to be an extremely important source of energy. We'll talk about this in chapter 3, which is going to be our next chapter. But this is where we get the majority of the energy to make ATP, and ATP is the primary energy currency within the cell. So glucose, in particular, is very, very, very important. So here are our monosaccharides. So remember, I want you to know the monomers for each and then the polymers for each of these macromolecules. Monosaccharides are the monomers of carbohydrates. These are the basic building blocks of all of our carbohydrates. We have 3 monosaccharides: glucose, fructose, and galactose. Now you'll notice they all 3 have the same chemical formula, C6H12O6. The big difference is going to be organization. So you can see glucose and galactose look very, very similar. Fructose, not so much. Instead of having a 6 carbon ring, it's going to have a 5 carbon ring. Okay, and then the big difference between galactose and glucose, of course, is going to be just organization in this case, not even so much functional groups. They have roughly the same functional groups too. Glucose is going to be the most important of these carbohydrates because this is going to be what we metabolize to produce ATP. Even when we eat or drink things like fructose and galactose, we're going to have to change those into glucose for the most part before they can be used for energy production. So we see glucose everywhere. It's very, very common. Fructose is usually found in fruit, and galactose, you can see the lacto part in that, which means milk. So this is going to be the basic building block of milk sugar. This is the only of the macromolecules where I'm going to ask you to give me something other than a monomer and polymer. You also need to give me what are called disaccharides. Okay, so this base word here, saccharide, means sugar or carbohydrate. Monosaccharide was 1 sugar. Disaccharide means two sugars. Okay, and so we're going to combine those monosaccharides we just saw into disaccharides, and they're going to give us different types of sugar here. So when we add glucose and fructose together, we now have sucrose. And sucrose is just regular old table sugar. Regular old table sugar. Okay? We're running into issues here because of the amount of fructose that we are consuming. We're getting a lot of fructose from the corn industry, and the body, our bodies really aren't doing well with this. So you'll see a lot of these. I find it slightly ironic and humorous, but like a lot of the soda companies are now going, oh yeah, we're just using pure cane sugar. Because they found that this cane sugar, this pure sugar, not that it's good for you, but it is better for you. Your body knows what to do with it. It's much better than these high fructose corn syrups. Okay, so glucose and fructose come together to form sucrose or table sugar. Just as another brief aside here, when we look at dental cavities, what are known as dental caries, before sucrose was consumed, really in any amount, there really were no dental cavities. There were really no. I mean, there were other problems with the teeth, but there weren't any cavities, and it was cavities rose the same time that the consumption of sucrose started to rise, and so if you really want healthy teeth, the best thing you can do for them is to eat less sucrose, eat less sugar in general. We have bacteria that crawl all over our teeth that actually eat the sucrose, and one of the byproducts of that fermentation of the sucrose is it produces an acid, and the acid eats away at the enamel, which allows other bacteria to get in and cause your cavities. So you want healthy teeth, don't eat sugar. The other disaccharides we have, if we combine glucose and galactose, we now have lactose, again, just another form of milk sugar. And if we add two glucose molecules together, we get maltose. Now, all these disaccharides, in order for them to be useful, we will have to break them down into their monosaccharides, so that we can break them down further and make the ATP out of them. Okay. Most of the junk food that we eat contains either monosaccharides, like we discussed here just a few moments ago, in the form of fructose, or disaccharides in the form of sucrose, which is the glucose and fructose together. But here's what's going on. So here's our glucose molecule, here's our fructose molecule, and you recall when we build things, we have to pull water out, right? So there's our water between these two functional groups. We pull the water out, and now this allows these two molecules to stick together. They're going to form a bond, right? So this is a dehydration or synthesis reaction. And now we have a disaccharide made from glucose and fructose that we call sucrose. So we've seen the monomers, the monosaccharides, we've seen the disaccharides. Now we need to look at the polymers. In the case of carbohydrates here, the polymers are going to be what we call polysaccharides. So again, that root word saccharide means sugar. Poly means many. So here we are going to have long chains of these monosaccharides. So here we can see, here on the left would be an example of glycogen. Here we have a whole bunch of glucose molecules. I mean, look at all of them. So these are really big and long chains. So this is why it gets the name of polysaccharide. There are two big forms that I want you to know here. The first type is going to be starch. And starch is going to be the storage form of glucose in plants. So over here, this is going to be starch. We can use, as humans, we can use starch as a major energy source. And so if you're, if you need some quick energy, something like a potato, a really starchy plant, could be very, very helpful to you. But again, I mentioned this. I can't remember exactly where I mentioned it, but I did mention it before in this lecture, where what we have to do is we have to take. So let's say we consume a potato, tasty potato. Um, this right here, this big starch molecule is much too big to be absorbed through the intestines. And so a big chunk of the digestive process is to break these bonds and cut these into individual monosaccharides, into individual glucose molecules. Okay, now the individual glucose molecules can get in, and then we can take this starch that is the storage form of glucose in plants, and we can use the glucose either for our own cellular processes, or if we're going to store it, we can take this glucose and we can store it now as glycogen. Glycogen is the storage form of glucose in animals. So the body doesn't like to waste anything, and so what we're going to do is if we consume an excessive amount of energy in the form of glucose here, then we're going to store some of it. We're going to store some in our skeletal muscles, and this makes complete sense as we'll talk about in our muscle lecture. Skeletal muscles use a lot of ATP, a lot of ATP. And so for normal function of the skeletal muscle, we want to make sure that we have enough glucose available to make the ATP we need for long-term energy use. Okay, so we're going to store some of this glycogen in the muscles. The other major area that we store glycogen is going to be in the liver, and so glucagon is going to act on primarily our liver cells when we have hypoglycemia. When our blood sugar levels are too low, we're going to use glucagon to release glycogen that was stored in the liver. Now the big problem is when the liver stores get full, we take that extra glucose and we're going to turn it into fat, and we're going to store it in the form of fat. So this is where we get the idea of these low carb diets. If you're not eating carbohydrates, then you're going to break down fat first or next, okay? You're going to break down fat, and you're going to use your fat as your energy source. But as long as you keep feeding your body carbohydrates, then this is going to be the first thing that it uses. It's because it's the easiest to use. We won't get into the biochemistry, but just straight glucose is actually really, really easy to make ATP from. Whereas if you have to use fat and you have to use other things, there's multiple processes where we have to change it and eventually change it into glucose or other molecules to put it into our metabolic cycles. But this is the idea: is if you're not eating carbohydrates, then you're not really then you can't use those carbohydrates for energy. You have to use other energy sources. Now this last one here I want to talk about is cellulose. Cellulose is a major structural component of the cell walls in plants. Now humans, we do not have the enzyme. There's an enzyme called cellulase. Remember that ending ASE usually refers to enzymes. So we don't, in our DNA, we don't make the enzyme cellulase. So when we eat cellulose from these plants, we can't break it down and we can't use it as an energy source. There are certain animals that can, and that's you look at something like cows, that only live off of plants. They have enzymes that can help them use these plant, this carbohydrate here, cellulose, for energy. But humans, no, we don't have the enzyme to break this down. So, which is also why when you have a salad instead of a hamburger, for instance, it's going to help you lose weight because you can't use any of the cellulose in the cell walls of the plants for energy. So you're consuming a whole bunch of food that you can't actually use, and so this is what we term fiber is cellulose here. So if you have foods that are high in fiber, you feel full because you're putting something in your stomach, but you're not actually going to be metabolizing it. You're not going to be breaking it down and using it for energy, and so it just passes right through. Okay, so many of you may have heard that eating celery, for instance, it takes more energy to eat it than it does than what you get out of it, and to a large extent that is true. Celery is basically cellulose with some water in the cells, along with some vitamins and minerals. And so when you eat a stick of celery, it expends more energy to chew it and break it up and to release the digestive juices and make the digestive juices than you actually get from it, because the vast majority of the carbohydrates, the cellulose, we can't break down. Now everyone thinks they're so healthy when they eat celery, but they always cheat, right? So because nobody really likes celery, uh, at least not plain, right? You basically are just using celery as a spoon for your peanut butter. Um, but if you were just to eat celery, you would, and please, I don't recommend this. You would lose weight rather quickly because you're just not getting very much energy from it. You, your life would be absolutely miserable, and there are other ways and better ways of losing weight, but that would certainly work. Moving on now to lipids. So remember, carbohydrates had a ratio of 1 carbon, 2 hydrogens, 1 oxygen. With our lipids, we're just going to get rid of the oxygens. And now we'll just look at the carbons and the hydrogens. And so it's going to be pretty close to a 1-to-2 ratio, carbon to hydrogen, with a little bit of oxygen sprinkled in here. Most of these lipids are going to be hydrophobic. Okay, water phobic, fearing. So we call them water fearing. They don't interact with water. And you recall water is a polar molecule. A lot of these lipids are going to be nonpolar, which is why they don't interact with the water. The lipids are going to be very, very energy dense. So they contain about 9 calories per gram, or I should say kilocalories. I should correct that, but 9 kilocalories per gram, compared to about 4 kilocalories per gram in carbohydrates and proteins. So very, very energy dense. So if you go look, for example, I was looking at a can of peanuts the other day. A quarter cup of peanuts, which is not much, like a handful of peanuts, had almost 200 calories. And you look at that and you go, well, geez, that's a lot. I could eat, almost a whole bag of popcorn and get just a little bit more calories than I would from this handful of peanuts. Well, peanuts have a lot of fat in them, so they contain a lot of calories per gram because of the amount of fat. There are going to be 5 classes of lipids. We're not going to talk about all of them. For example, eicosanoids. This is the only time we're going to talk about eicosanoids. But there are 5 classes of lipids here. We have our fatty acids, eicosanoids, our glycerides, steroids, and then we have our phospholipids / glycolipids, where we take lipids and we start adding other things like phosphates or sugars. So let's start with our fatty acids. Fatty acids are long chains of carbon with a carboxyl group on one end, okay? And then we're going to have lots and lots of hydrogen associated. Remember, we talked about how we have carbon associated with hydrogen. So look, almost every one of these carbons has 2 hydrogens attached to it. This one's got 3. This one doesn't have any on the end. So it's almost a 1-to-2 ratio of carbon to hydrogen. But we have this long, long chain of carbon with hydrogens attached to it. Now, these fatty acids can be described as either saturated or unsaturated. And when we talk about a saturated fatty acid, you'll notice that each one of these carbons has a single bond between them. Okay? And then what that means is there is as many hydrogens as possible attached to this long chain of carbons. And so every one of these carbons has 2 attachments to hydrogen. With an unsaturated fatty acid, we can see here we've now got a double bond between 2 adjacent carbon atoms. And because of that double bond, you also notice that we are missing 2 hydrogens. So this is no longer saturated with hydrogen atoms because we now have 2 missing. Okay, so that's going to be the big difference between saturated and unsaturated fatty acids, is the presence or absence of those double bonds, and then the presence or absence of those hydrogen molecules. Now, this does make a difference in how we metabolize and how we use these fatty acids. One of the big challenges, you've probably heard this, that we want to limit the amount of saturated fatty acids we get, and it's true because of this these single bonds and the saturation with hydrogens. They tend to form this really straight type of molecule, which we can see here. This is a great picture. And the problem with this is these straight long fatty acids, they tend to stack on top of each other very easily. Okay, and which makes them very difficult to metabolize. It makes it more difficult to break down. Sorry about that slight problem, but yeah, so they're very difficult to break up. They stack in there nice and tight, um, and they tend to clog things, like arteries. Okay, so we want to if we want a diet where we have more unsaturated fatty acids than fatty acids. You can see when we add this double bond that it puts a kink in this long chain here, which we can see right there. This nice little kink. Okay, and so they don't stack up nice and neat like our saturated fatty acids do, and so they're easier to metabolize, and they don't tend to clog arteries and things like that. Now, there can be, this right here is an example of a mono unsaturated fat because we have a single double bond. And we could also have a polyunsaturated fatty acid where there are multiple double bonds. And so many of you are probably familiar with fish oil. Fish oil is a polyunsaturated fatty acid, and it tends to be much healthier for us than our saturated fatty acids. Fish oil is anti-inflammatory. It kind of helps keep the cholesterol levels in check. There are a lot of benefits to fish oil. If only it didn't, come from fish, which I think is kind of gross. So usually the big difference here between saturated fat, other than the carbon, but we're talking about sources. The source difference between saturated fatty acids and unsaturated fatty acids. Most saturated fatty acids have animal origins, and unsaturated fatty acids usually have plant origins. Our glycerides. So our fatty acids, they can't really be strung together like our monosaccharides or our amino acids, which we'll see here in a little bit. But they can bind to glycerol, and so glycerol is going to be the backbone of our glycerides, which makes sense here. So we can have a monoglyceride, diglyceride, or triglyceride. We're probably most familiar with the triglycerides. But all this means is we have our glycerol backbone, and if there's only one fatty acid attached, then it's a monoglyceride. If there's two, diglyceride. If there's three, then we have our triglycerides. These glycerides are going to be formed using those dehydration reactions. You can see we're going to pull water out there, water out there, water out there, and we're just going to stick these fatty acids directly to the glycerol backbone. Okay. These triglycerides have many really, many important functions. Go back and think about the adipose tissue in our histology lecture. And the adipose tissue, what were some of the functions of it? Well, energy. Okay, we can use these. These are fatty acids. We can use them for energy. Insulation, protection. So they surround and protect organs. Those types of things. They are going to help body contouring. These glycerides. So most of our adipose tissue. Remember, you'll have an adipocyte with a large triglyceride droplet inside. Our next category of lipids are going to be our steroids. So steroids do have what we call a 4-carbon ring structure. You can see 1, 2, 3, 4 carbon rings. And then we have just different functional groups attached to this basic ring structure, which is going to make the difference between these different steroids. So a lot of these different steroids are very close, minor changes in these functional groups. So 2 examples of steroids here would be estrogen and testosterone. Testosterone is the primary steroid that is going to be produced by males. Does that mean males don't produce estrogen? That's not what that means. Males do produce estrogen, but the primary sex hormone is going to be testosterone. Women, we have one of the primary ones is going to be estrogen. Does that mean women don't produce testosterone? No, that's not what that means. It's just that the testosterone levels are much, much lower. But if you start looking, there's a lot of similarities between testosterone and estrogen. There's just very minor changes. Okay, we're missing we're missing a double bond here and here. We've changed it here. But it's just going to be these functional groups on the testosterone that are different than the functional groups over here on estrogen. The other really important steroid that I want to mention here is cholesterol. Okay, and now you can kind of see sterol, right? Steroid. Okay, cholesterol. Cholesterol is got a bad name in medicine, and which I partly understand, partly don't understand. But the body makes its own cholesterol, so. If we're consuming more than we need, then the body's just going to make less. So, I mean, I don't quite see the worry about it, but cholesterol is going to be very, very important in strengthening our plasma membrane. So please remember this, because when we talk about it in the next lecture talking about the cell, I'm going to mention this. So it's going to help strengthen. It's going to help give the plasma membrane some rigidity, and then it, like I said, it just helps strengthen it so that it doesn't burst open and kill us all. So cholesterol is another very important steroid, which we can see up top here. Again, very similar to our other steroids here, estrogen and testosterone. Big difference there. Look at that functional group that's hanging off the edge of cholesterol. But we're going to see lots and lots of cholesterol in our plasma membranes. Last slide here on lipids. We are going to look at our phospholipids and glycolipids. The big one we're going to talk about here is phospholipids. Phospholipids are extremely important because this is going to be the basis of our cell membranes. Again, we'll talk more about this in chapter 4, but our cell membranes are going to be made up of it's a phospholipid bilayer. And all we have is we're going to have these fatty acid tails. Okay, these are going to be this is going to be the lipid part. Okay, we have these fatty acid tails, and then we have a phosphate head. Now, when we look at a glycolipid, it's the same thing, but instead of having a phosphate head, we now have a sugar head. Okay, so here we can see we've got three sugars kind of attached. But let's focus on the phospholipids here. So we describe these fatty acid tails as being hydrophobic. The phosphate head is actually going to be hydrophilic. Philia means to love. So if you look at Philadelphia, its nickname is the city of brotherly love. Okay, so that's kind of what I always think of there. So hydrophilic means it loves water. And so what's going to happen here, as you can see, this is a great example of this micelle. So made up of phospholipids. All the phospholipids kind of arrange themselves on the outside. The phosphates on the outside, and then the hydrophobic tails on the inside. And this is the same basic setup we're going to see with our phospholipid bilayer. This allows the phosphate heads to interact with the water while keeping the hydrophobic tails away from it there. So we've talked about carbohydrates, we've talked about lipids, and the next macromolecule on the list here is our proteins. And they are the most abundant of all of the macromolecules. Proteins are everywhere. Remember those enzymes that we talked about earlier in this lecture? Those are all proteins. We talked about proteins when we looked at the histology. Remember, the collagen fibers were made up primarily of the protein collagen. The elastic fibers were made up of elastin. The reticular fibers were made up of collagen. So collagen is the most abundant protein in the body, and proteins are the most abundant of the macromolecules. The basic building block of all of our proteins, the monomers, are going to be amino acids. And we're going to talk more about the amino acids here shortly. But let's just, as we're introducing proteins here, let's talk about the functions that all of our proteins are involved in. So support structural proteins. We just talked about collagen and elastin and many, many other structural proteins, actin and myosin in the muscle cells, okay, are going to be there for movement. We have some proteins that are going to help with our buffering. We talked about buffering. We talked about acids and bases. Transport proteins in the blood. Hemoglobin is our major oxygen transport protein. We also have other proteins in the blood that are there for things like osmotic forces and things like that. Um, Metabolism. I just talked about love enzymes. You know I love enzymes. So enzymes are going to basically drive all the metabolism, all the chemical reactions that occur in the body. Coordination and control. So when we talk about hormones, there are two general kinds of hormones. We have our lipid hormones and we have our protein hormones. So fat and protein hormones. They're going to act differently, but many of the hormones in the body are going to be protein based. Defense antibodies are going to be proteins, immunoglobulins, which are these antibodies. We also have keratin in the skin. We talked about that as well when we talked about the epidermis. We'll talk about it more a little bit later, but keratin it's waterproof, but that thick layer is going to help protect the body from invading microorganisms and things like that. So we have lots and lots of functions of these proteins and lots of different types of proteins. Now the monomers, as I mentioned on the previous slide, are amino acids. And there are 20 amino acids, and they all share a very similar structure. We have a central carbon. Okay, we have an amine group over here, which would be the NH2. We have the carboxyl group over here, the COOH. We have this hydrogen attached to central carbon, and then this R group. So this area right here. This is what's shared by all of our amino acids. What's going to make each one of them different is going to be this R group, and that just means the functional group, if you will. And this functional group is going to determine all the chemical characteristics and properties of that specific amino acid. Okay. Now, of the 20, 9 are considered to be essential, and 11 of them are going to be non-essential. Now, really, I'm not a huge fan of the terminology here, just because it can be terribly confusing. What we say by essential, what that means is we can't make them. These 9 essential amino acids have to be consumed. They come from outside of the body. The 11 non-essential amino acids, they're all important, but the non-essential amino acids are produced in the body. We actually make them in the body. And so we don't have to go to outside protein sources for those 11 non-essential amino acids. Like I said, I think the terminology is incredibly confusing. The first thought is, oh, I need these ones. I don't necessarily need these ones. And it's wrong. We need all 20 of them. It's just some are produced by the body, so we don't need external sources, and others are not produced by the body, so we do require external sources. And one of the best complete proteins, you'll hear that term bandied about, complete proteins. And what they're talking about is a complete protein is one that has all 9 essential amino acids, and most of those are going to be animal products, meats and the like. Not saying you can't get the essentials from eating a plant-based diet. You certainly can, but it does take a little bit more thought and a little bit more planning. You can't just you have to have a lot of, if you're live vegan or vegetarian, things like that, then then you have to be very, very mindful of what proteins you are consuming and in what amounts to make sure that you're getting those 9 essential amino acids. Just makes it a little bit tougher, certainly not impossible by any means. Now, these amino acids, remember, these are the building blocks, these are the monomers. Okay, so these amino acids, they are going to come together to form the polymer. The polymer is known as a polypeptide. Polypeptide. Okay, so big long chain of amino acids, this is known as a polypeptide. The average protein, the typical protein, has about 1,000 amino acids combined, so 1,000 amino acids in length. Some of the larger ones can have over 100,000 amino acids, and obviously the shorter ones can be, as low as a couple hundred of these amino acids. But all we have is just this long chain of amino acids held together by what are known as peptide bonds. So this base word peptido, peptide, anytime you see that pep, you should automatically start thinking proteins and amino acids. Okay, so we have peptide bonds, we have dipeptides, polypeptides, all of them are about proteins. Okay, now we're going to see it more on the next couple of slides here, but this sequence of amino acids, okay, is going to influence the function of a protein. If we start swapping out or changing the sequence of amino acids, we end up changing or completely changing the function or rendering our protein non-functional. So the sequence of the amino acids here is incredibly, incredibly important. So I want you to start thinking here, we'll talk more about it here in a second, but I want you to start thinking, where do you think the instructions come from as to which amino acid is where in the chain? Kind of start pondering that. Think about that for a moment. Look at this. It's red, it's bold, it's all in caps. There are 3, count them, 3 exclamation points at the end. It must be important. This is something that you need to know and just stick it in your brain forever and ever and ever. The shape of the protein dictates the function. The shape of the protein is the function. If we change the shape of the protein, then we 100% change the function of the protein. I want you to think about those enzymes that we talked about previously. If you need to, remind yourself, go back and look at the pictures of the enzymes. But remember those enzymes, they had an active site where our substrates were going to bind. And we talked about the lock and key method. Now, what would happen if that active site were to change shape? Do you think those substrates would fit in there as nicely as they should so that we can complete the chemical reactions? No. So the shape of the protein is incredibly, incredibly important. If we change the shape at all, we change the function. And this, I'll be honest with you, I completely missed when I was in school. Learned about proteins, I can't tell you how many times, just like, why do I care? Why do I care what the primary, secondary, tertiary, and quaternary structures of this protein are? Didn't get it. Now I understand, okay? Because the structure dictates the function. So let's talk through these different structures, and then we'll go to the next slide. We'll look at some visual representations here. So the primary structure is the sequence of amino acids in the polypeptide chain, just like we discussed on the previous slide. So we have aspartame, and then we have, or not aspartame, leucine, and then right next to it is isoleucine, and then we have glycine, and then, so this is that sequence of amino acids. Okay, this is the primary structure. For each different protein, they're going to have a different sequence of amino acids. Now, I asked this question on the last slide, and hopefully you've put a little bit of thought into it, but where do the instructions come from that dictate this sequence? This is where our DNA comes into play. Okay, a gene is a sequence of nucleotides that codes for a specific protein. So the sequence of nucleotides in the DNA directly relates to the sequence of amino acids in the protein. Okay, so that's the primary structure, just a sequence of amino acids. Now what happens is that that long polypeptide chain, it starts to fold and bend in on itself, and that initial folding is known as the secondary structure. And these amino acids will start to make hydrogen bonds between each other. Now this is going to lead to what we call an alpha helix or a beta pleated sheet. So those are the two options there for a secondary structure. The tertiary, which means third, so the tertiary structure is where things just continue to bend, and we start making more bonds between the secondary structure. So that alpha helix starts to bend a little bit more, and now the whole alpha helix bends, and they start making bonds, and that's going to be our tertiary structure. In general, the tertiary structure is going to be the final three-dimensional shape. Quaternary structure. This is not every protein. Not every protein has a quaternary structure. Here, with a quaternary structure, we need two or more tertiary structures. So two or more tertiary structures are going to come together and form this gigantic protein complex, and that's going to be our quaternary structure. So let's go to the next slide and let's look at some of the pictures so we can kind of start associating these structures with what they look like. All right, so what was the primary structure? Remember, the primary structure was just the sequence of amino acids. Here's amino acid 1, amino acid 2, and so on, okay? So whatever that sequence is the primary structure of the amino acid, okay? Now here with our secondary structure, you can see A1, A2, A3, A4, and then there's a bend, right? So A5 bends around, and then A6, 7, and you can see now there's this dotted line right here. That is a hydrogen bond between A6 and A4, and another one between A7 and A3, and when we just kind of keep turning, okay? Here you can see a very similar, but instead of making this pleated sheet, is how they describe it, we make a helix. And here, again, dotted lines mean hydrogen bonds. So the secondary structure is just kind of the first interaction between these this polypeptide chain. From our secondary structure, we go to our tertiary structure, and here you can see like there's the alpha helix again. Here's another one, and so it just kind of, the whole alpha helix kind of starts to bend in on itself, and it makes this very, very complex three-dimensional shape. We don't quite appreciate the three-dimensional shape from this view because it's just a drawing of a protein, but very, very complex three-dimensional shapes. Our last structure is going to be a quaternary structure. And here you can see we have two purple proteins and two blue proteins. And they're going to kind of smash together, make this gigantic protein right here. And in this case, it happens to be hemoglobin. And that's going to be the quaternary structure. We have multiple tertiary structures that kind of come together. Here we have a collagen fiber. And you can see here we have there's going to be one tertiary structure. Here's another tertiary structure. So we have three different tertiary structures that are kind of wrapping around each other, giving us a nice long collagen fiber, which is pretty cool. Okay, so this is what we're talking about. And again, I can't emphasize this enough. This sequence of amino acid is going to end up giving us these final shapes that we're looking for. Okay, if we change the sequence of amino acids, I mean, you could change from like a hemoglobin shape to a collagen shape. I mean, probably not with just one amino acid change, but you get the idea here that this sequence of amino acids is incredibly, incredibly important. And that's what our nucleic acids are going to help us out with, is they're going to make sure we get the proper sequence of amino acids to make our proteins like we need them. All right. I think I mentioned this before, but just in case I didn't, I'll just mention it again. The final three-dimensional shape of the protein determines its function. So shape equals function. If we lose the shape at all or change the shape, then we change the function. Now there's a process here called denaturation where that three-dimensional shape is changed. Okay. Now this usually occurs when the proteins are exposed to heat or pH changes. Okay, so here we are. Here's our very complex quaternary structure protein, right? You can see the 4 different strands. Okay, and let's say we add heat to it, which we can see right there. And what the heat does is it starts to break all those hydrogen bonds that was holding this whole protein together. Now all those hydrogen bonds break, and the protein starts to break apart. Okay. Now if this was an enzyme here that was going to help us make ATP, all of a sudden we can't make ATP anymore because this protein lost its shape and can no longer catalyze the reaction that we need it to. Okay, so you've all seen this in play if you've ever cooked an egg. Okay, so if you look, and here you can see it. So when the egg first splashes in, the egg white is actually not white. It's kind of this pale yellow color. And it will stay that way unless we add heat to it. When we add heat to it, all of a sudden it turns from a liquid form into a more rigid, rubbery, hopefully you don't have rubbery eggs, but rubbery, and it turns white. What's happening there? All the proteins are being denatured by the heat from the frying pan. And that's how we cook an egg. That's how we cook our meat. Okay, cooking a meat, you are adding heat to it and it's denaturing the proteins and is changing the structural shape of those proteins. Okay, now this will usually, depending on how bad the heat is or the pH change, it will usually lead to irreversible damage. Not always, but can quite often lead to irreversible damage. So if you've ever wondered why a high fever is so problematic, here you go. If our body temperature gets too high, our enzymes no longer function like they're supposed to. Our proteins no longer function like they're supposed to. You can actually denature proteins in your brain, which can lead to permanent brain damage, all from a really high fever, high sustained fever. So it's anything over 104 is incredibly worrying, and needs to be, that individual needs to be cooled down as quickly as possible. Because otherwise irreversible damage can start to occur. Now we always talk about heat, but one of the things I do want to mention here is the pH change. So I've never been able to try it because I'm not a fish person, so the thought of this is just gross to me. But many of you may have heard of ceviche. So what ceviche is this is a fish dish, and I try to say that without smiling, but I can't. It's a fish dish that is chemically cooked. So instead of cooking the fish on a stove top or a grill or something like that, they take this cold fish and they put usually it's lemon juice. They put lemon juice all over it. That acidic lemon juice causes denaturing of the proteins in the fish, and so it chemically cooks these proteins. Like I said, the thought of this is just gross, that you have this cooked fish that's still cold and covered in lemon juice. Like, yeah, just that's not going to happen for me. Many people love it. I've never been able to try it, but this here is the same thing is happening to our egg and our fish here, just different methods of denaturing the protein. One is using heat, and the other is using pH. So let's finish this strong. We only have just a few more slides. Let's talk about our nucleic acids. These are the last of the macromolecules we want to talk about. The basic building block, the monomers of our nucleic acids are nucleotides. Okay, and this I'm really big on this. Please make sure you remember what are the monomers and what are the polymers for each of these macromolecules, if we mention them. Okay, now each nucleotide is made up of 3 parts. We're going to have a phosphate group, Then we are going to have a pentose sugar. Pent means 5, so we have a 5-sided sugar. And this pentose sugar is going to determine whether we are looking at DNA or RNA. DNA stands for deoxyribonucleic acid. And so the sugar there, unsurprisingly, is deoxyribose. Remember that ose end usually means a sugar, so deoxyribose. RNA stands for ribonucleic acid, and its pentose sugar is no longer deoxyribose, but just ribose. So ribonucleic acid has a ribose sugar. Deoxyribonucleic acid has deoxyribose as its pentose sugar. Okay, so that's going to be a change from DNA to RNA. So phosphate group, this pentose sugar, and then we have a nitrogenous base or a nitrogen containing base. And there are going to be 5 different nitrogen containing bases that we'll look at on the next slide. So the 5 nitrogen containing bases, okay, and I don't care here about purines versus pyrimidines. Please don't memorize those or put those in the category. I really don't care. Okay, but just for the sake of organizing my slideshow, I put them in there. Okay, but I won't ask you that those on the exam. So we have the 5 nitrogenous bases. We have adenine, guanine, cytosine, and then the next 2, it depends on if you're looking at DNA or RNA. If you're looking at DNA, then the 4th one is going to be thymine. If you're looking at RNA, the 4th one is going to be uracil. So these 2 are dependent upon whether or not it's DNA or RNA. Now, the absolute most important thing that I want you to know for nucleic acids, and I'll mention this again, I think it's in the next lecture, is what we call complementary base pairs. I really should put that in red. I forgot to put that one in red there, okay? Very, very important. Adenine is always going to bind with thymine, okay, because we're talking about DNA. In RNA, adenine will always bind with uracil. Guanine will always bind with cytosine. And this is going to be extremely, extremely important. So learn this now so that when we talk about transcription and translation in our next chapter, it makes more sense. But this is the absolute key to nucleic acids. When I look at how DNA and RNA function, it's so simple and absolutely beautiful in how it functions. It's amazing to me, with an infinite amount of complexity associated with it. But when you look at just the basics of how it all works, it's absolutely simple and beautiful. And it all comes down to these complementary base pairs: adenine and thymine, guanine and cytosine. Okay, now the backbone, so what we refer to as, so we always look at again, I'm just going to focus on DNA here. We always look at DNA like a ladder, okay, and the rungs of the ladder are going to be our nitrogenous bases. Okay, so here's the rungs. So we have adenine binding with thymine because A always binds with T. So that would be the rung of our ladder. And then the rails of the ladder, the sides of the ladder, are going to be made up of these alternating phosphate, sugar, phosphate, sugar, phosphate, sugar. Okay, so the rails of the ladder, phosphate and the sugar, the rungs of the ladder are going to be our nitrogenous bases. Okay, and we can see it right here. What holds us all together? Those magical hydrogen bonds. You didn't think they were going to be important when I said, they're weak bonds and they're transient bonds, but they're incredibly important. Look, these hydrogen bonds are holding our proteins together. They're even holding our DNA together, which is really, kind of important. Okay, so that's going to be the basic structure of DNA, of our, I shouldn't say just DNA, of our nucleic acids. Even with RNA, we have a same setup, we just don't have the double helix. So RNA is going to be a single strand, DNA is a double strand. So you'll hear it called the DNA double helix. RNA just has a single strand, so no complementary strand on the opposite side. But you can also see, this is RNA because you can see the uracil. And this is DNA because you can see the thymine. Now, what is the primary role? Why do we have our nucleic acids? Their primary role is to store and transfer information. Specifically, they're going to store and transfer information to make proteins. There are some other things they make, but that, like, if you learn nothing else about this, that's what I want you to know, is they store and transfer information to make proteins. Okay? So remember that that sequence of amino acids was incredibly important to the overall structure and shape of the protein. We get that specific sequence from the sequence of nucleotides in our DNA. Okay? Now, a lot of times when we think of DNA, we think of inheritance, and that's absolutely true. But what do we inherit? We inherit proteins, if you will. So if you have brown eyes or black eyes or blue eyes or green eyes, okay? And it all comes down to the different types of protein that you are producing. And those proteins, or the instructions for those proteins, are going to be passed. They're going to be inherited from your parents. So yes, they do control our inherited characteristics, but that's not the only thing I want you to think of. Is remember, they are holding the information for proteins. Okay. We talked about DNA, we talked about RNA. The last thing I just want to mention here before we end this lecture is there are 3 different types of RNA, 2 of which we are going to talk about in our next lecture. The third one here, rRNA, is ribosomal RNA. This is going to help us make our ribosomes. So we'll talk about ribosomes in the next lecture, but we really won't talk about rRNA. mRNA is messenger RNA, and I want you to think of this as these are the directions from the DNA. So from our DNA, we make messenger RNA. tRNA is called transfer RNA, and it's going to transfer amino acids to the messenger RNA. And we'll dig into that quite a bit more deeply in the next lecture when we talk about, again, transcription and translation. But that's it for chemical principles. A lot of important things in here, a lot of just basic things to help us figure out what's going on in the body, not just this semester in 121, but please remember a lot of this because you're going to mention it again in 122 A&P2.